Hello, everyone. My name is Kelly Hall. I'm a marketing coordinator for ALS North America. I want to thank you all for joining us today for our Webinar Wednesday series. Our presenter today is Dr. Roy Radcliff. Roy received his Bachelor's of Science degree in Animal and Veterinary Science from West Virginia University in 1993, his MS degree in 1995 from Michigan State University, and his PhD in 2001 from Michigan State University. He spent 2.5 years at the University of Missouri, where he developed a real-time PCR assay to quantify growth hormone receptor mRNA in bovine liver tissue. In 2004, he joined Marshfield Clinic Laboratories Food Safety Services, conducting research focused on pathogens and pathogen detection methods and diagnoses of John's disease in cattle. Over the past 10 years, Roy has held the titles of Laboratory Manager, Chief Science Officer, Director of Applied Sciences, and Chief Executive Officer. Now, Roy serves as the Director of Operations for USA Food Testing for ALS Environmental. Today, Roy will be discussing current and future pathogen detection methods in food. Feel free during this presentation to use your chat box, ask a question, or raise your hand features to ask questions throughout this webinar. Roy, thank you for taking your time to deliver this presentation today, and I will go ahead and turn it over to you.
Thank you, Kelly. Hello, everybody. Thank you for joining the webinar today. One of the most important things to consider when choosing a method is if it is fit for purpose. I don't know of a single method that works great on every food matrix. Some examples are, some food dyes can interfere with the fluorescent detection used in some of these methods. Some food matrices have proteins or minerals or compounds in them that can interfere with the enzymes, such as like Taq polymerase, which would inhibit the DNA amplification and ultimately the detection. So evaluating a food matrix and having data to support suitability is important. Sometimes this data is available from either the scientific literature or the manufacturer of the test kit. If not, then most likely you would need to perform some sort of validation on your own or with a lab capable of doing such validations. So those validations will include accuracy and sensitivity. Now, all the method manufacturers will do some sort of inclusivity and exclusivity analysis to evaluate whether or not the method will cross-react with closely related organisms, whether it's specific to the organism that it's targeting. But there's no way for them to test every organism, and there's no way for them to test every different food matrix. So a lot of this is done through spiking of your food matrix or particular food matrix in predetermined sample sizes. But ultimately, when it comes to pathogens, the goal is to be able to detect one organism per the desired sample size. And over the years, the sample size has increased. A lot of initial manufacturer validations are done on 25-gram samples, but we find ourselves now testing samples anywhere from 100 to 375, upwards to maybe even 1,000-gram samples. The second factor to consider is the time to result. How fast do you need this answer? And for this, you need to ask yourself a few questions, like, do you hold your product until results are in? If so, how long is the shelf life of your product? And ultimately, balancing your production with storage and shipping all play a role in determining how fast you need the result. If you have a 5 to 7 day shelf life and it takes 2 days for a result, and you're holding product, that only leaves you 3 to 5 days to get it delivered to its endpoint and its time on the shelf. The third thing to consider is the cost. And cost has two major components. The cost of the analysis, which is a direct cost, and that has several factors within it. The cost of the kit and the consumables, the cost of any special equipment that's needed to perform that test, and then the cost of the expertise that's needed to perform that test as well. Some technologies require very little expertise, and some require a high degree or high level of expertise in order to interpret. And then there's also some indirect costs or costs not associated with the actual analysis that should be considered. Is there lost time on store shelves due to holding the product? If it requires refrigerated storage, what is your cost to hold that product in a refrigerated state? So these are a few things to consider when choosing a method. For the rest of the presentation, I would like to talk about various technologies used to perform the actual pathogen detection. The very first technology I'll talk about is culture. Historically, culture has been the gold standard. And as the gold standard, all new technologies and methods are compared to it during the validation process. Using various medias for pre-enrichment or selective enrichments, differential plating and identification, it can be very organism specific and ultimately get down to the exact organism in which you're looking for. And regardless of whether you're using a culture method or any other of the methods I'm going to talk about today, for a screening method, if you want to perform a confirmation, ultimately it will come back to a culture method typically. Now, regardless of the screening method you choose, there's always some level of enrichment. Either a two-step enrichment with a pre-enrichment with a selective enrichment following it, or a single step which incorporates both the pre and selective enrichment by adding some inhibitors to hold back background organisms and allow the targeted food pathogens to grow. So although culture is probably the most definitive and reliable, culture methods take a lot of time. We have to grow a microscopic organism to the point where we can actually see it with the naked eye. And they are very often complex. Depending on the organism, there could be several tests that have to be performed. And the result of one of those tests can trigger one or more additional tests, which then take additional time to grow. They also require interpretation. Is it a dark blue colony or is it a light blue colony? Is it producing gas or is it not producing gas? Even though there are predetermined outcomes to these questions or tests, there's always exceptions to the rule. Every culture method will tell you what the typical colony should look like, but if you don't see typical colonies, then they say you should also continue on with what they call non-typical colonies. Manufacturers have made advancements in culture technology to try and make things easier. One of these is dehydrated films, where the culture media is dehydrated and pre-prepared. These are designed to save time. With the addition of selective augers or selective medias to this, you can also speed up the process and do your primary enrichment and a selection at the same time. The picture on the screen shows a film that's designed for detection of E. coli and coliform. And then there's also the biochemical tests, the tests that were used originally, to really narrow down and to a specific organism. And these have been simplified with a panel type test. And they bundle several of these enzymatic tests together, which result in various color changes. And depending on the color change, there's a code assigned to each well. And then that code can then be decoded to give you a specific organism. Other advancements in culture is combining some of these biochemical tests into the selective augers. Single medias, they save time with only having to plate the organisms or the sample once. It also reduces the needed expertise to interpret. In some of these cases, it's becoming a lot easier to distinguish various organisms, such as Listeria species from Listeria monocytogenes in the example on the left, or E. coli and Salmonella, the example on the right. So in effect, it's performing both a selective growth of these organisms as well as a biochemical differentiation of them, and allows for the rapid differentiation of these organisms. So the next evolution of detection was enzyme immunoassays. Every living cell has hundreds, if not thousands, of proteins on its cell surface. And this technology detects these proteins using specific antibodies that locate and attach to the proteins on the cell surface. Now, the key to this technology is finding an antibody that detects and attaches to the protein that is only found on the surface of the organism in question. The next step is being able to detect when this antibody-protein interaction occurs. And this is usually done with a second antibody attached to some sort of detector. An example of this would be a horseradish peroxidase enzyme. These two antibodies effectively sandwich the cell protein or the antigen in between them, and this triggers a reaction that can be seen with the naked eye. Now, these are commonly performed on an absorbent pad or strip of some sort. As the sample absorbs through the strip, it interacts with the first antibody and then the second antibody. And if the target antigen is present and the sandwich is between the two antibodies forms, then a dark line will appear on the absorbent strip. Now, these are only as good as the specific antibody or the specificity of the antibody to the antigen and how unique the antigen is to the cell in question. Many organisms share a lot of antigenic properties. So there can be some cross-reactivity with an antigen attaching to a cell that's not the intended target. And then there is the interpretation of it. Is there a line or not? It can be very subjective. If there is a weak signal, somebody with the naked eye has to make the call. Is that a line? Is it not a line? Because sometimes the line is very faint. In this case, we're forced to make a call. And then from the laboratory side of things, we're going to make a conservative call. And if we think there's any sort of line, we're going to call it presumptive and recommend that you continue on to a culture confirmation. An advancement to try and take out some of the subjectivity of the human element in this, or the interpretation, is the use of a fluorescent detector instead of something like horseradish peroxidase. In this case, the second antibody is replaced, the detector on the second antibody is replaced with a fluorescent indicator, which are not normally visible to the human eye. So in this case, we rely on a machine to detect and measure that fluorescent and make the call. And if the fluorescent is strong enough, then the instrument or the machine tells you that it's positive or presumptive positive. Now, these use the same antibody antigen technology. It just removes some of the expertise needed to interpret and replaces it with a machine and a computer. Additional advancements with the enzyme immunoassays is the use of phage technology. This improves the selectivity and the specificity of some of these enzyme assays because they utilize the phage tails, which have evolved over billions of years to their specific host. They've evolved to find that one specific cell type that they target for their own replication, and this natural selection process is used to improve the selectivity of these assays. Now, these technologies generally use an engineered tail fibers from these phages to identify and capture the target organism before the second antibody is added for the detection phase. Using just the tail fibers and the engineered tail fibers prevents the laboratory from becoming contaminated with live phage. So, you know, if we're trying to detect salmonella, we have a live phage in the laboratory that targets live salmonella, it could become very difficult to detect it if the phage is destroying the cells before we could ever grow them to the point of detection. So by using genetically engineered tail fibers, the actual live phage is not present. It just using the tail fibers to select and capture the target. And over the years, we've advanced from looking at the proteins on the cell surface to actually detecting the genetic material used to encode those proteins. So in this class of technologies, I refer to as genetic technologies. PCR was one of the first genetic technologies used to target specific and unique DNA sequences. The key is to find a DNA sequence that's only found in your target organism. The sequence must be unique, and then probes must be used to target that sequence to allow the DNA or the PCR reaction to occur and amplify the target DNA. If the genetic sequence and the primers are not unique and specific to that organism, the accuracy of the test is compromised. Sometimes this is not easy with closely related organisms that have very unique or very similar DNA sequences. So once you have the specific sequence for that organism that you want to target, and you've designed probes to replicate it, how do you detect it? We're still dealing with microscopic endpoints. So in order to be able to detect it, we have to replicate that sequence so we can visualize it with our naked eye. The sequence or amplicon is then copied over and over again using an enzyme called Taq polymerase. By heating and cooling the reaction tube over and over again, the Taq polymerase attaches to and copies the target sequence. So in theory, if a reaction is 100% efficient, each heating and cooling cycle would double the amount of the target sequence that we're after. So to give you an idea of how fast this ramps up, if we start with one copy of DNA and we run the cycle, heating cooling cycle once, we end up with 2 copies. 2 cycles give us 4 copies. 3 cycles give us 8 copies, and so on, doubling with each cycle until eventually we have billions of copies of DNA. So even with billions of copies, we can't see it with the naked eye. So the initial technology was that we would take that dye or take that DNA, combine it with a fluorescent dye, and run it out on an agarose gel and visualize it under UV light. If the target sequence was present, it would produce a line as seen in rows 2, 3, and 4. And if the target sequence was not present, it would be blank, such as line 5 or row 5 in the picture on the right. So as with all technologies, there's advancements. And the incorporation of the fluorescent dye into the reaction and to make the detection process easier in real time. Now there's several dyes and many different ways to use them to detect the target amplicon, all with varying degrees of sensitivity and specificity. The least specific dye used in a lot of PCR-based assays is cyber green, and it binds to all double-stranded DNA. And then there can be very specific probes, and there's several different kinds of probes used that could also be used to detect the target sequence. These probes are then tagged with dyes, and some of them even have quenchers, so the 2 probes have to be present, or the probe has to bind in a specific configuration in order for fluorescence to occur. The other advancements is the refinement of the Taq polymerase and the technologies that's used to heat and cool the sample over and over again. With newer technologies, this heating and cooling cycle can be sped up. So instead of minutes for each cycle, it takes seconds. It makes it very fast. And it would reduce the actual test time from 3.5 hours down to 60 minutes on the machine. And then once you combine the detection method of the fluorescence, you can actually take a measurement after each cycle and then actually have a real-time indication of whether the amplicon or target sequence is being amplified as the test progresses. Another twist on the PCR technologies is digital PCR. And really, it's just miniaturization of the PCR reaction. Your PCR mix is separated into many, many different small reaction wells, and they are run all at once. Now, I have very little life experience with this technology, but the claim is that by decreasing the volume of the reaction, you actually reduce the inhibition on the Taq and allow more efficient amplification. And because of the small reactions, you can get to the point where you have like a single copy in each one of these mini reactions, and you can ultimately do some sort of quantification of the number of cells in your end product. Now, this is all dependent on how many copies of that gene are present in the genome. If it's a single copy and you do get single pieces of DNA in each well, it would be very easy to do the quantification. Other advancements is isothermal PCR. So instead of using heating and cooling to separate the double-stranded DNA for replication, a DNA helicase is added to the reaction, and it's used to separate the double-stranded DNA and allow the Taq to replicate the target sequence without all the heating and cooling cycles. This is very rapid. It can go to completion in as little as 15 minutes. It's very conducive to running small batches. You don't need as much capital tied up in instruments, and samples aren't tied up on a machine, or a machine isn't tied up with an analysis for 1 to 3 hours. So when you combine that with the fluorescent technology, you only need some sort of reader of some sort to detect and measure the endpoint fluorescence to determine if your target sequence was amplified. So the next advancement in PCR technology is transcription-mediated amplification. So we've gone from targeting proteins on the cell surface to the DNA responsible for encoding some of those proteins. And this technology actually targets the cellular machinery, specifically the RNA used to build these proteins. The advantages of this is that a single cell that would have, say, one gene encoding a specific protein may have up to 10,000 ribosomal RNA molecules within it to build those proteins. So instead of targeting one intracellular target, you now have 10,000 targets. And with more targets, you can reduce the enrichment time and actually get a result quicker.
Hey,
Now,
Roy.
since RNA is, yes,
Roy, this is Kelly. We have a question that came in regarding that from Joe. I'm going to read you his question.
sure.
He says that I understand how PCR works. The DNA gets denatured and becomes 2 ssDNA. The forward primer attaches itself to one strand and the reverse primer attaches itself to another. My question is, how does the primer know when to stop synthesizing?
Well, there's various ways for that to happen. In the initial reactions and some of the early PCRs, there was no stop point. It just ran until the heat cycle completed, which stopped the replication. So your initial amplification products may actually be longer than, say, 200 base pairs. But as you continue on cycling, eventually those become shorter and shorter. I hope that makes sense, until they ultimately get to the point where you're only replicating between the forward and the reverse primer.
He says, thank you.
You're welcome. So with the TMA, or transcription-mediated amplification, one claim is that, RNA is less stable than DNA and degrades much faster. And so the claim is that you're more likely to detect living cells versus dead cells, because once a cell dies, the RNases, not only within the cell but within its environment, quickly degrades any RNA. So if you take the TMA technology and you combine that with the isothermal technology and the use of fluorescent probes for real-time detection, you can have a very specific, very fast detection method. Another technology on the genetic, in a genetic grouping, as I will put it, is the whole genome sequencing. And whole genome sequencing is a big buzzword right now in the pathogen detection area. There's been a lot of advancement in speed, and with more options available for this technology with various providers, the price is coming down. However, as a screening tool, I personally, or initial test, I really don't see this as a future for WGS. The power of whole genome sequencing comes in its complete specificity, completely comparing the entire sequence of the entire genome of the organism, and being able to use that and compare it to other known organisms and use it for tracking and tracing genetically identical organisms throughout a plant or in products. And it's a very powerful tool. The predecessor to this might be considered a lot of the pulse field gel electrophoresis or SNPs, comparing other, ribosomal products. And this is such a powerful tool that the government is now using it to tie specific organisms to illnesses, and back to products or even plant environments. So it's a very powerful tool for being able to track and trace organisms through your products and through your plant. The last technology that I'd like to talk about is what I consider molecular-based technologies, and really there's one major one out there right now, and it's the acronym is MALDI-TOF, and MALDI-TOF stands for matrix assisted laser desorption, ionization, and time of flight. And basically, we're using mass spectrometry, and it's been modified so that it can be used to identify organisms. The downfall to this is you do require to get to an isolate for this technology. So as a screening technology in a mixed culture, it's not real viable. You still have the time and expertise needed to get to a specific isolate. Once you select that isolate, you place this into the instrument or on a plate in the instrument, and then a laser is used to basically vaporize those cells. And all the various molecular components from those cells are then detected and graphed using mass spectrometry. So like mass spectrometry, this profile is then compared to a known library to generate an ID of the organism. This technology can easily distinguish between genus of organisms. And in some cases, you can get to the species level. And eventually, I believe this will replace the biochemical ID for confirmations.
Roy, we have another question from Joel. He says,
Right.
does this have the ability to determine whether an organism is alive or dead?
Well, in this case, you actually have to grow and cultivate the organism and isolate a colony. So you would only have live organisms to put on this instrument. All the pre-cultural steps in order to get it to a single colony on a plate has to be performed ahead of time in order to be able to use this to identify whether it's E. coli or Salmonella or Listeria. So it would only be able to detect live organisms.
Thank you.
Some of the more recently, a few companies are now combining some technologies for various advantages, either to improve speed or ease or reduce cost. In some cases, the PCR technology, instead of being coupled with fluorescence and real-time detection, is being coupled with EIA type technology. And then once you do the PCR reaction, you place that onto, say, a lateral flow device and combine it, and look for a line to form on the absorbent pad to determine if your target sequence was there or not. And in some cases, this same immunotechnology is being combined with liquid crystal technology. for both speed and ease of use. In this case, you still have antibodies that are coated onto microspheres. They bind to the antibody or the protein or the antigen or the protein on the cell surface, and then run through a liquid crystal. Light is then passed. If there is agglutination of these antibodies and cells, it disrupts the light flow through the liquid crystal, and you're able to detect that change in light with a detector on the back end. So when it comes down to deciding which technology is best for me, there's hundreds of methods available to screen your products or your environment for pathogens. Narrowing them down to which is best can be daunting. We've not gotten to the point where we're to say like Star Trek tricorders, where we're able to just wave something across our sample and say, yeah, it has E. coli in it or not. In the end, there's several methods that are very robust and work on many different food matrices, but it really is customer and product specific when it comes to choosing a method. It goes back to, is it fit for purpose? Will it work with my product? Does the turnaround time meet the needs that I have for this? And is the price point where we can afford it? And then, sometimes we've had clients come to us who say, I'm selling this to company X, and company X requires that I use, this specific method to test for the, salmonella or listeria. So which test you choose is dictated by the end user of your product. But in general, there's rules of thumb. Newer methods are getting faster and faster and more specific. But as with anything new and faster is generally more expensive. And depending on the method, the level of expertise needed varies as well. Typically, the less expertise that's required to perform it, the more expensive it could be, because you're tied up in complex equipment or detectors needed to do that interpretation. So if you need assistance evaluating which method best fits your needs, we have the experts that can help do that. Everything from determining what organisms you should be looking for to which methods may work best for your matrix. There's a lot of times we may already have validation data for a specific matrix. If we don't, we can help you generate that data. And then, through discussions, figure out which methods can meet your turnaround time needs and which method works best for your situation. And with that, I'll gladly take any other questions, and I thank everybody again for joining today.
Thank you, Roy. Yeah, we'll go ahead and open it up for questions. Feel free again to use the chat box, ask a question, or raise your hand feature, and we'll go ahead and get those questions started. As we are waiting for those questions to come in, I do want to thank everyone for attending the webinar today. This has been recorded, and we will be posting this on our YouTube channel, where you'll find a wide range of video resources. This will also be posted on our alsglobal.com website. Just search Webinar Wednesday to find this recording and past Webinar Wednesday presentations. You can also follow our LinkedIn page as we post links to future webinars and also other valuable resources. We don't have any questions yet, but we'll just give it a couple more minutes for everyone to write in their questions or raise their hand. No questions yet. We do have a comment from Joel. Joel wanted you to know that as soon as you develop the tricorder, he'll buy one. And also there was,
that would allow me to retire right away.
well, then we don't want that to happen yet. Also, Joel asks, will we get a copy of this presentation? We do email all the attendees today the presentation next week. And again, we also post this on our YouTube channel and our alsglobal.com website. We have one question coming in from Joel again. He says, do the methods that test for the surface antigens also ensure that the organism is alive or dead?
Typically, I would say yes, because of the pre-enrichment needed in order to get them to the point where we can detect them. Typically, the only time we really have issues with detecting dead organisms is with some of the genetic-based methods, and if there is just an enormous amount of that organism present. So in general, I would say that the enzyme assays would most likely only detect live organisms. The issue you have there is some of the specificity associated with them. Some are much more capable of distinguishing similar organisms, and I think one of the organisms that EIAs have struggled with in the past has been the close relationship between, say, Citrobacter and Salmonella. Some of them have a hard time distinguishing the two. And if you have a product, going back to history, say sprouts or sprout water, where we had a lot of issues one time with what we would call a false positive because we were detecting Citrobacter in the sprout water instead of actually Salmonella.
And Joel had a follow-up question because he's been asking these questions about the live or dead organisms. He did want to follow up and ask, is this an actual concern?
Detecting live versus dead?
Yes.
Again, it would probably be situation specific. If you're monitoring like an environment, a drain or something, versus a product, you would definitely only want to be able to detect live cells in a product that you're selling to the public.
Okay, and it does not look like there are any additional questions. Roy's email is listed there in case you have some follow-up questions. Joel, feel free to send him an email directly with your specific questions. Other than that, again, I want to thank you, Roy, for the presentation today, and thank you to everyone for attending, and have a wonderful, wonderful rest of your week.
Thank you.